Welcome again, everyone, to today's lecture on digital twins, intelligence, and plasticity. I will cover the first hour of this talk and will mostly talk about intelligence, while my colleague Luca Brandt will cover the second hour of this lecture and he will talk about plasticity. So you know about the work we are doing in the brain simulation section of the Charité. We build brain network models. For this, we put people into MRI scanners to measure their structural brain connectivity, so how brain regions are connected via these fiber bundles that we saw here. And we divide the brain into a set of brain regions, and then we aggregate these fiber bundles into a compact representation, a connection weight, and a time delay to arrive at such an abstract brain network model. So as input, we have diffusion MRI, tractography, and reconstructed surfaces. And this gives us a structural connectome-based brain network model. And the model then, when we turn it on, it produces dynamics. And these dynamics produce then functional connectivity matrices. So these are the matrices on the right-hand side. Here's the laser pointer. uh, which are the fitting target of the brain models. So we have our empirical functional connectivity measured in the MRI, and we have the output of the model. And now we tweak the parameters, we tune the parameters in order to maximize this fit. This is the target of the simulation. So to get from brain data to digital twins, and you have heard this already in the two first sessions by Petra Ritter and Leon Martin. As input, we have diffusion-weighted MRI and structural MRI to create the model. This gives us then functional MRI for tuning the model. And we use this in this specific study that we are talking about today to explain behavior, to come towards mechanistic theories of how brain produces intelligent behavior. And we did this with the specific study by the Human Connectome Project, the young adult study. So here there are young subjects, 1,200 in total, and they undergo extensive testing, not only neuroimaging and MEG, but also behavioral testing, intelligence tests, and all kinds of tests are being made with these persons. All the results of this work are published in this article. Feel free to read more. I'm putting the link into the chat where all the details are. Um, so, intelligence is a notoriously difficult topic to talk about. It's really hard to define what is intelligence, and there's always a lot of often emotional discussions around intelligence. This definition is works quite well, I would say. It's by one of the leading intelligence researchers in the field, Deary, who analyzed the data of really hundreds of thousands, if not millions of persons regarding the intelligence. So his definition is, and this was agreed by, as we can read here, by 52 prominent researchers on intelligence. Intelligence is a very general capability that, among other things, involves the ability to reason, plan, solve problems, think abstractly, comprehend complex ideas, learn quickly, and learn from experience. It is not merely book learning, a narrow academic skill, or test-taking smarts. Rather, it reflects a broader and deeper capability for comprehending our surroundings, catching on, making sense of things, or figuring out what to do. Intelligence, so defined, can be measured, and intelligence tests measure it well. So, this was now a definition, but it probably gives us not really an intuitive understanding or really a mechanistic theory of what intelligence really is. I bet every one of us from youngest age on wonders why is another person more intelligent, so to say, more fit? Why do they have better grades, for example, in school? Or nowadays, we might also ask, what distinguishes human intelligence from AI, from ChatGPT, LLMs, transformers, and so on? Um, and we now would stepwise come to a neurobiological explanation. And we start by these definitions here. Solving a complex problem often involves a recursive decomposition into easier subproblems. In computer science, this is often called divide and conquer, and this is a widely used strategy for coming to decisions. So here a hierarchy of decisions, of decision-making processes is formed, and this then requires that all these subproblems in this hierarchy, so we have a big problem to solve and we decompose it into simpler problems, a whole tree of problems to solve. So this requires that this hierarchy is held in working memory until the solutions from problems lower in the hierarchy arrive to be integrated into the overall solution, which is the opposite of jumping to conclusions. So you not immediately have the result in mind without thinking, but you need to think. And this means solving subproblems and integrating the results of subproblems into an overall solution. And put this way, intelligence can be understood as a scale-free fractal process that depends on multiscale excitation inhibition balance, because excitation inhibition balance modifies the timing with which information is integrated to form a decision. So this is now very deep already into the theory we are about to learn, which we'll cover now. This took the gist away, but of course you didn't understand it now, but we will now work towards this understanding. So we now already heard that there are different types of decisions to be made, different types of problems to be solved. There are easy and fast problems like interpreting a traffic light, right? You do not need to think at all. When you drive with your car or your bike and suddenly the traffic light turns red, you do not need to think at all. You immediately know what to do. And this doesn't require any longer form of intelligence. On the other hand, if you drive with, here's the Berlin subway map, if you need to find the way from A to B on such a map, this is often not so easy. To find the best route, you need to, decompose this into smaller problems. What train do I need? Where do trains run? And then, walk along this way and merge the outcomes of these finer, simpler problems into your overall solution. And this takes time and is slow. So, as we have already heard from prominent intelligence researchers, there is a general factor of intelligence, the so-called chief factor. So there is a whole lot of different types of intelligences. I bet you have heard of emotional intelligence, for example, and so on. And indeed, there are different types of intelligence, different tests, vocabulary comprehension, for example, information processing, here arithmetic, picture completion. So there are many different types of tests that could be made to measure cognitive ability. But the interesting outcome of these tests is that individuals who perform well in one cognitive domain tend to perform well in all of the others. And this then forms the general factor of intelligence, the chief factor. So as you can see here, G strongly correlates with all these different submetrics like reasoning, spatial ability, memory, processing speed, and vocabulary. And we can also see in the lower panel that age strongly correlates, or in this case, mostly an anticorrelation, with these faculties. So intelligence decreases as we age, except for vocabulary, which has a positive correlation, as we can see here. So we can also distinguish fluid versus crystallized intelligence. Fluid intelligence is much more, actually, I should have put this on the right side. It's much more related to the harder, the longer taking problems. So this is the ability to solve novel reasoning problems, and this is associated with comprehension and problem solving. So you cannot learn to solve this, you must work out the solution as you go, basically. Very similar to the subway map, where you have to work out the solution. And here tests usually have little cultural or scholastic content. It's nothing that you need to learn, but it's something that you need to figure out. And Raven's progressive matrices are such tests. I bet you have seen them already, and we will see them in a minute. And this contrasts with crystallized intelligence, which is more like one-shot intelligence, more similar to what an LLM is doing, I would say. So this reflects acquired knowledge and skills. So this is something that was learned and is now deeply ingrained into our brain network structure, and just is just fired out when it's required. And this depends a lot on education, cultural background, and so on. And here tests um, also include scholastic and cultural knowledge, such as vocabulary. But Raven's matrices, they really test for fluid intelligence. As you can see here, these are two tests. The left one is simple. We don't need to think really, right? So we have here three rows. They're rectangles, and we immediately see, the rectangle is always tilted 90 degrees to the right. Same in the middle row, the circle is tilted 90 degrees to the right. So we can conclude also in the lower row, then the filled square is on the right side. But if I now look on the right-hand side to the other test, this is a hard test, or a hard Raven's matrix. So I really would need to now look very carefully at these symbols, and maybe start counting dots, and really start thinking to find a solution. So as you can see, we can form a continuum of simple to hard problems with these Ravens matrices, which will become important when we look at the results of our 1,200 subjects from the Human Connectome Project study, which we'll come to in a minute. But there's also another important correlation with intelligence, and this is processing speed. And I bet many of you also had this pet theory, and often people think, and this is one of the classical and traditional theories, probably someone else is smart because their brain is faster. This is some theory many form informally, and also science forms formally. And it has strong support. This was shown over and over again since a century basically already. And meta-analysis over 172 studies with 50,000 participants shows this robust correlation. It's really remarkable. These processing speed tests are super simple. Press a button when the color green appears. Split-second decision, right? This is a super easy and quick solution, but nevertheless, it correlates very strongly with intelligence, with all other intelligence metrics. So processing speed and reaction times, they correlate strongly with all other faculties. And this was already first demonstrated in 1890. So this was like the first intelligence test, so to say, published in the journal Mind in 1890. So these were the very first intelligence tests, and this was shown now over and over again, and also shown over lifetime, that there is this strong association between processing speed and the other intelligence metrics, with the exception of vocabulary knowledge, which also tends to increase. But here we can see how all others decrease together, and speed is closely linked and correlating not only with intelligence, but processing speed is, this was also a famous study, closely associated with death and disease. So when we create such models here and look what is the explanation causes of death in studies where a lot of biomarkers and behavioral metrics are collected of people, then the strongest association, or rather the second strongest association. Smoking is a bigger killer, but on second place already is the reaction time as the strongest explicant of impeding death, basically, even before physical activity, blood pressure, resting heart rate, and so on. All these typical indicators of disease and impeding death correlate less than this super simple magic reaction time, processing speed. So there's strong support for the idea that reaction time is some proxy of bodily system integrity. It has something to do with how well we are and how well we function, how intelligent we are, how smart we are. But is it really the causal factor underlying intelligence? Is speed of our brain, so to say, really the cause that makes us smarter or not? And there are important counter arguments, and also the study we are presenting here is a counter argument to this, because training processing speed does not transfer at all to other cognitive domains. You can become faster and faster and faster, but this doesn't make you smarter. To solve simple problems, we need mental flexibility. So in contrast to solving hard problems, where we need to keep the solutions in working memory, the sub-solutions, the sub-problems, if we do not have a pen or a computer to write things out and revisit them later, we need to store them online in our mental scratch pad, our working memory. Um, so for simple problems, we need the ability then that decisions do not rely on extensive accumulation of evidence. So we want to quickly shift. We cannot like look for a minute at the traffic light to really be sure whether it's red. We need to act immediately. But this is different for harder problems, of course. There we need to keep them in memory, keep them online, to have them available, so that the evidence from these solutions or decisions can then accumulate and used for other decisions. So for solving complex problems, the timing matters. So to now solve the problem, we need to make, as mentioned, this recursive decomposition into simpler problems and then make decisions about these solutions and then integrate it higher up in memory. So decisions need to be held out until sufficient evidence arrives to not prematurely jump to conclusions. If there is insufficient information, our brain will just jump to a conclusion, and this conclusion may be more often wrong than right. So we are not the first ones to come up with such a theory. There are similar theories out there for a while. For example, this very influential theory by Nobel laureate Daniel Kahneman, Thinking Fast and Slow. And many of you surely have heard about this book. And he has this two systems, basically. System one operates automatically and quickly with little or no effort. So this is our fast mode. And system two allocates attention to the effortful mental activities that demand it, which is often associated with the subjective experience of agency and choice. And mistakes occur if we allow system one, the fast system, to process a cognitive decision, while we should have really passed this decision over to our system two. And this is a great book, a great theory. I bet Daniel was a great man. Unfortunately, he died last year or 2 years ago. But we now are going to recast his theory, thinking fast and slow, into decision making fast and slow, because we believe this is the underlying mechanism. And we found that participants with higher intelligence were only quicker when responding to simple problems. So actually, we now turn this century-old observation since 1890, as you have seen. This was the state of the art. It was assumed that higher processing speed means you are more intelligent. But now we found actually an important exception to this rule. In our data, actually, participants with high intelligence were only quicker when responding to simple problems. They needed more time to correctly solve complex problems. And this became apparent in the 24 Ravens matrices questions. So these were sorted from simple to hard and got increasingly harder. So processing speed tests, as you know, are very simple, but with fluid intelligence, there is no time limit. So people, the requirement is really to think until the solution is there, and this, yeah, then leads to longer time, of course, and the requirement for more thinking. So when we look at the data now very explicitly, so here we have the general intelligence in this column. Here we have the fluid intelligence in the second column. In this row, in these two rows here, we have two typical processing speed tests, and we reproduce the typical century-old observation. People with higher intelligence have a higher processing speed. So there's a positive, highly significant correlation. But now we were surprised to see the correlation between the reaction time for the fluid intelligence test and intelligence. We also see here a positive correlation, but these times here, these are real times, while this is the inverse of time. So here a positive correlation makes sense. This is a speed measure, the inverse of time, but here we have time. This is seconds or 2 minutes, and they correlate positively with general intelligence and with fluid intelligence. So this means actually the century-old observation is not true, at least for hard problems where you need to think. So we thought about the unexpected. So this is unexpected, and we thought this is a trivial explanation. So fluid intelligence tests are arranged in order of increasing difficulty, and they are discontinued after five incorrect answers. So it could simply be that the people with a higher reaction time, simply because they advanced farther in the test, where the questions got harder, they took longer. So of course, yeah, one could argue because the intelligent people took more time to solve the problem, to really solve the problem, this took longer. But when we now look at the reaction times of individual test questions, so really the reaction time for every one of the 24 individual test questions and correlate this now with the intelligence of the test subjects, we see a very specific picture. We see that up to a certain, so here on the x-axis we have 1 to 24, the 24 questions, and on the y-axis we have the correlation. And we see from question 1 to 10, the usual pattern is reproduced. People with higher intelligence are faster. So here's the higher intelligence, they need less time. But for here now it's reversed. At the higher end of the questions, when the questions get harder, suddenly the people who are more intelligent, who solve more test questions correctly, also needed more time to solve them correctly. And interestingly, also, those people had a higher resting state FC. So there's a stronger correlation between the slow solvers. So there's a strong correlation between the reaction time. Here we have the reaction time for the fluid intelligence test on the x-axis, and on the y-axis, we have the FC, the average FC, the average correlation between brain areas. So FC again is the functional connectivity, how strongly all brain areas correlate with each other. So this is the average over this whole matrix summed up. And here we can see that regardless of the complexity of the question, there's always a strong correlation between the reaction time and functional connectivity. So slowness seems to be associated with intelligence. Slowness seems to be associated with functional brain networks. So now we want to understand what is the underlying mechanism? How do brain networks reconfigure to be slower or faster and make fewer or more or fewer correct decisions, basically, and fewer errors or more errors? And to do this, we augmented brain network models. So in the past, brain network models usually only had these forms of connectivity that we see in gray here. So there was connectivity, long-range connectivity between the excitatory populations, and there's local connectivity between excitatory and inhibitory populations. But what was absolutely missing in brain network models was the connectivity from excitatory to distant inhibitory populations, which exists in the brain. And now we add this. Now we, for the first time, incorporated this in brain network models. And this allows us now to control excitation inhibition balance. Excitation inhibition balance is an important topic in neuroscience and computational neuroscience, especially because it can be associated with so many cognitive functions and also pathologies. So here on the left-hand side, we see what a high excitation inhibition ratio means. So there's strong connectivity between the excitatory populations, and there's only a little connectivity towards the inhibitory populations. And here we see what a low excitation inhibition ratio means. So the inhibitory weight is very large, while the excitatory weight is very low. So here we can see this again if we define EI ratio as the quotient of long-range excitation, so the long fiber bundles that go between excitatory populations um, above FFE, the feed-forward inhibition. So here we have also long-range connectivity, but locally this excitatory input is then converted into inhibitory input for this local excitatory population. So this is long-range excitation over feed-forward inhibition, which gives us excitation-inhibition balance. And the cool thing is now we can start to tune this. Now we can take this parameter EI ratio and start tuning it instead of only tuning a long-range excitation, often called also the G factor probably in the first two lectures, and instead tune this full ratio. And this gives really rise to very interesting dynamics. So here we see again our simplified brain network model, and we start, as you can see, with a very low excitation inhibition balance. So there's a lot of feed-forward inhibition and not much long-range excitation. So this is a lot of yeah, inhibition here. And here we see then the time series of these two populations, the two excitatory populations. So we see how these time series unfold as we now change the excitation inhibition balance from low to high, the excitation inhibition ratio. And here we already see the outcome of this video here. We will then tune when we increase the AI ratio, the correlation, the functional connectivity between these two time series from a high negative value crossing zero towards a high positive value. So let's start it. And we can see here how the time series change and undergo a complex intermediary state and then end up with full correlation again. So we can smoothly tune here from total anticorrelation, here we can see how they are anticorrelated, to full correlation. So we can fine-tune synchrony now. We can smoothly tune synchrony, the synchronization of brain areas from anti-synchronization to full synchronization via a critical complex intermediary state. And this was for me like the holy grail of computational neuroscience, the relationship between structure and function. How SC relates to FC. This was like really the big question I started out when I started my PhD. This was the holy grail. Well, how does brain dynamics, brain activity result from brain structure? And all that was needed was to add feed-forward inhibition. This was just missing before. And this allows us now to smoothly tune really from full anti-synchronization. We can see how here the time series are, the peaks and troughs are on opposite ends, to full synchronization via this complex intermediary state where chaotic and complex activities happen, which is also a common pattern. These are all complex patterns. These are all known patterns in the brain. And now we can smoothly tune between these patterns from low to high. And all based on very little assumptions. This is all very simple. All we did really was take four oscillators here and add this connectivity here between the excitatory and the inhibitory oscillators on the long range scale, and then smoothly tune this. So this is nothing. There's no complex dynamics that were injected into this. And this leads me to believe that this could even be a fundamental principle across all scales. There's no reason why this principle should break anywhere. It could happen, this could happen anywhere, from down to the quantum scale, up to the cosmic scale, basically. As soon as there are excitatory and inhibitory actors and they are connected, these dynamics emerge. This can be shown with our computational study. And not only can we tune the relationship between just two nodes and two entities here, so you might ask, okay, this is probably very simple for tune nodes, but as we increase the number of nodes and three-body problem with three bodies already, dynamics become very complex. So usually, surely it breaks down when you add more nodes. No, it doesn't. Now we will tune the full brain network model. So here we have roughly 400 brain areas. And here we have the empirical functional connectivity, our target. And we start out now with the simulated target. So this is the fresh brain network model without any change, just turning it on based on the normal structural connectivity. So now if we start the tuning, what we do now is what I have just shown here, what we have just seen, this tuning of a single pair of brain areas, we now do simultaneously between all pairs of brain areas. So you will see now all these lines go bigger and thinner. So the algorithm now varies them. And I started, and you can see um, the algorithm now varies these connections. It varies the excitation inhibition balance, and the fit dramatically increases. Such fits have never been there before. Normally, a fit of brain network models, the usual FC fit that you get out of a brain network model is maybe zero point 0.5, zero point 0.6 if you're lucky. But now we can perfectly fit brain network models. It's a correlation of 0.999. There's no discrepancy between the empirical and the functional brain network. That's what that we simulated with our brain network model, our computer model. And the algorithm is super simple. There's nothing, I have revealed the whole story already to you. So we can form an algorithm out of this simple intuitive concept of excitation inhibition balance and the ratio of long range excitation and feed forward inhibition. So all we do is really we simulate a few time steps, produce a new functional connectivity matrix, and then we compute the difference between our empirical and our simulated functional connectivity matrix. And when we notice that in our empirical functional connectivity matrix, there is more correlation between two brain areas than in our simulation, then we increase EI balance in our simulation. Vice versa, if we notice that in our empirical matrix there is not enough correlation, not enough synchrony in the brain between a pair of brain areas, then we decrease the correlation and move towards antisynchrony. And we do this until we have the desired fit. And we were able to do this for all the 650 subjects from this study, which had complete data. And as you can see here, compared now so this was the state of the art before. So before, if you tune these subjects, then you get a correlation of something around zero point 0.3 maybe, or here on average it was 0.24, which is a typical result of brainwork model simulations in the past. And now we approach here one basically perfect correlation. And when we now can fit the brain network model so perfectly, we can now start asking what actually happens inside the brain network model, what mechanisms are at play here that could help us understand how these differences between people intelligence tests come to play. And what we see here is that as we can tune functional connectivity from low to high, we can also when we tune the EI ratio from low to high, also the input correlation of synaptic activity has basically the same shape as our functional connectivity. This is, yeah, this can be really expected. This is nothing like super surprising, right? If fMRI measures the strong correlation, then we can also expect in the electrical activity and in the synaptic activity similar forms of correlation, and this was shown here. But the actual mean amplitude shows a much more interesting shape. So this shape here is much different, and the input amplitude behaves much differently than the correlations. And this will then give us the explanation for the intelligence differences. Well, we can move on here. This is just this. So now we ask what happens in the brain model that could help us understand these intelligence differences. So which aspects in the simulations, so not empirical data anymore, in the simulations now correlate with the cognitive performance. And indeed, so this is the reaction time of the fluid intelligence test. We have the strong correlation between the input correlation, the correlation between synaptic input currents that go into decision-making populations, and we have a strong negative correlations with the amplitude. So if you imagine the activity, the input that goes into a population of neurons is some oscillating curve, but it has a mean value, right? And this mean value shifts depending on excitation inhibition balance, and this will help us understand the behavioral differences. So with a higher FC, again, we have a decreased amplitude and a higher synchrony. And we now systematically explore this in our working in our winner-take-all model. So this is a very specialized circuit that allows to make decisions. So our usual brain network models, they are also very similar to the type of populations that we use here. But these populations have the ability, so they have cross inhibition, as you can see here. So there comes evidence for option A. So now we need to make a decision, right? We now need to form a solution for a simple problem. Is it A or B? There comes evidence for A here, evidence for B comes here. If more evidence for A comes here, then this excitatory population will, or this pair of excitatory inhibitory populations will inhibit these other B option populations here, leading to recurrent excitation and ultimately a successful decision for outcome A, because A got a little bit more evidence and was able to inhibit the other outcome more, and this led to the decision then. As mentioned, these equations of the model of the decision-making circuit and also our normal brain network model circuit, the reduced Wang-Wang, are very simple. It's basically the same equations with slightly different parameters, and this allowed us then to actually couple the large-scale brain network model with this decision-making circuit. So as you have seen here, we have now implanted in posterior parietal cortex and in prefrontal cortex um, a AA and BB. So basically A in PPC and B in PPC and A in PFC and B in PFC. So there are two times these populations in the prefrontal cortex and parietal cortex, and they are connected also to the large-scale network. So instead of noise, which was the usual driver for the decision-making circuit, we now have the large-scale brain network input as the driver. Just very quickly, because we are running a bit out of time, very quickly how these decision-making circuits work. So as you can see here, we have in blue and violet populations A and B, and now evidence comes in and evidence is being accumulated. And as soon as one of the two populations crosses a threshold, so this is happening in parietal cortex. As soon as the threshold is crossed, in prefrontal cortex, the decision is made. We have a quick ramping up of activity here in prefrontal cortex. And this was a fast decision. Here in blue, we have a slow decision. So here we can see how slowly over time, evidence is gathered until finally the the boundary was crossed, and here also prefrontal cortex activity ramps up. So we have in violet the quick and fast decisions, and in blue we have the slow decisions. So we can see how it takes time, that evidence comes in. Here's a region where it's not really clear, will A win, will B win, and then A really makes the race, and the decision is formed for A. Very similar also how working memory is represented in the brain. This is ongoing activity. So here this orange is an active working memory, then this blue is a twist factor trying to inhibit it, but no, it quickly came back online. It was just briefly inhibited, but immediately came back. While here was an arrow trial, there came a distractor, and the orange activity immediately died out. So this online activity that would be needed for working memory to persist was destroyed in the second case. So now we can study working memory and decision making in isolation first before we couple it into our large-scale brain network model. So here we have excitatory inhibitor populations, and we then inject now excitation inhibition balance. We simulate now our brain network model and again tune from low to high. And importantly here, there is of course this correlation then. So these decision-making populations A and B cannot be assumed to have uncorrelated inputs, which was also in previous model a problem, I would say, which we have now improved here upon. So we can now tune um, both of these features that we have identified as potential mechanisms that allow us to understand the behavioral differences. On the top row, we have amplitude. In the bottom row, we have the correlation between populations as the defining feature. So we tune here the amplitude from low to high in PFC and here in PPC. And we see that when the amplitude decreases here, the number of correct decisions increases, and also the integration time. So perfectly reproduced what we know from empirical data. And very similar here, if we increase the correlation, then the number of correct decisions increases, and also the integration time increases until a certain point and then decreases again. So we have now gone full circle. In the empirical data, we have seen that slower subjects found the better solution and they had a higher functional connectivity. In the large-scale model, we have found that a higher functional connectivity comes along with a decreased amplitude and an increased synchrony. And now in the small-scale decision-making and working memory circuit, we have seen that a decreased amplitude and increased synchrony leads to slower but better solutions. So here we have now the explanation, and we can now also study it with so-called phase-space models. Um, Here we see the phase space between population A in PPC and population B in PPC. So this is now in parietal cortex, and this is now the race between the two populations. And this is the default state. This is how how the model um, would normally actually. No, this is not the default state. Let's first look at the video and then we go to another slide which shows it better. So you see how these circles here move. So let's just start this again. These circles here are so the circle up here and the circle down here are attractors. They pull the populations to decision A or B. While here on the separating line here in the middle, the outcome is open. So we can see that with the default amplitude, this is the middle here, there is just this normal. Let me just stop this video to a good point. I don't have controls here. That's a pity. But we can see partial. I'm a presenter mode and I cannot control the video, unfortunately. But we see what I wanted to show here basically is that with the lower amplitude, the slower decision making, we gain a new attractor down here. This is a low activity attractor. And this allows us to collect evidence. This gives us the time to collect evidence and not immediately ramp towards one outcome. So um, one so this new attractor here allowed us to keep the system in a low state and allowed us to integrate evidence. On the other hand, with increased amplitude, now we see actually the default state. So this is the default state. We have only a separating. This filled circle means the activity is pushed quickly towards one of the two options. But when we now increase the amplitude, you will only see this very briefly. A new unfilled circle emerges at this place. And this allows to keep both options online. So this is like our very active state where we keep both options open to immediately react, like the tennis player swaying left and right to being able to immediately react. So maybe we see it when you look quickly. Yeah, have you seen it? There was quickly this attractor, but it got quickly merged and pulled downwards. So we moved from high to low input. So even if you didn't understand the full details of it, don't worry. This is a very complex topic, and I try to cram now a very complex topic in just 1 minute, basically. The point is, with these phase-space diagrams, we can now find a mechanistic explanation with attractors, separators, saddle points, and so on, and bifurcations that allow us to understand this in in the language of dynamical systems and dynamical systems theory. And the same thing we can also do with noise. So if you remember, we previously have seen that the correlation of the input amplitudes, of the input currents, um, leads to slower decisions and higher decision accuracy. So now we replace this noise component from the equations I have shown earlier with correlated activity. So we can study how the correlation of activity impacts the decision-making behavior. And but we needed to invent something new to do this because phase portraits in the past they didn't really actually consider noise. So phase portraits are usually from the perspective of a deterministic system without any noise. So you have a unique gradient vector, a unique arrow that points into one direction. But with noise, it's not an arrow anymore. It's more like such a glyph that we see here, pointing predominantly in a certain direction, but sometimes also in other directions. And these glyphs can get rounder, so there's no real dominant direction, or they can get more elongated, where there's a more circular flow, a more laminar flow. And so here we can see this in this video. So here we start with relative circular flow here. It's very round. There's no clear preference here in the middle. But now when we will increase the correlation, so we start with zero correlation, this is perfectly uncorrelated, and we can see this here in the middle part, it goes in all directions. While if we already start out with quite a lot of evidence for for option A or B, then we would anyway be quickly pulled to attractor A or B. But if it's not so clear, if the problem is hard and we really need to collect evidence, then with a low correlation, we will just, we don't really have our option to stay close to this diagonal, which keeps us open, but it goes into all directions. And now if I start the video and you see how the correlation increase, you will see how these glyphs in the middle start to get elongated, and this keeps us open. This keeps our brain open for input to process new evidence for any of the two options. Let's start it. Yeah, so as you've seen with the increasing correlation, it gets we get more and more drawn to this center diagonal, which keeps the mind open, so to say. So we have here the no correlation, medium correlation, high correlation um, uh, differentiation. I'm just showing you these videos to give you this picture. In the comparison directly. We can see here how this leads to much better outcomes. We have here all green correct solutions for the high correlation, and with the no correlation, we get much more faulty results. And this can be easily explained by the nature of noise. So if noise is correlated, of course, then the the vectors point along the diagonal, while if it's uncorrelated, it points into all directions, basically. And we can also make some points about working memory. So a decreased amplitude makes working memory more robust, so less likely to override a working memory state, makes it more flexible. We can quickly react to the red traffic light, which is sorry. The opposite is the case. With a decreased amplitude, the working memory is more robust, and we can now keep sub-solutions in mind while remaining parts of the brain compute other solutions to form this hierarchy and to integrate all sub-solutions into an overall outcome. While a higher amplitude allows to quickly override working memory states and makes us more responsive to new stimuli and the red traffic light. And yeah, when we now couple the small scale circuit with the large scale brain network model, we see that indeed the brain network models of our 650 subjects, they do what their empirical counterparts do. So these are really digital twins. The brain models of smarter subjects made more correct decisions and took more time for decision making. And now coming towards the end, just some validation and robustness checks of the AI tuning algorithm. So to really, one could say, is this really a unique solution that the algorithm finds? And it's so we did resimulations. We did this fitting, this AI balance tuning with 1,000 resimulations with slightly different initial conditions. And we found a little bit of a jitter, so parameters were not exactly the same, but the resulting time series, the FMI time series, were exactly the same. So the algorithm always pulls the brain into this comparable mode which allows us to make this analysis. And also, all these mechanisms that I have shown, they are stable over resimulations when we vary the noise seed and the initial conditions and everything that can be varied. And basically, we get coherent results out of this. So, my last slide to wrap this up now, the summary of the first part of this lecture. Intelligent people take more time to solve complex problems, but are faster to solve simple problems, as was long known. Brain network models can be fitted with arbitrary functional networks based on a biological plausible fitting algorithm, the excitation-inhibition balance. The dynamics of the fitted models relate to cognition of the subjects, supporting inference of healthy and pathological neurocomputational mechanisms. And by coupling decision making and working memory circuits with brain network models, we can now simulate high level cognitive processes. We have now task virtual twins that allow us to explain the intelligence of the underlying humans. And we see this as a first step to really artificial human intelligence. And it also shows us now coming back to Daniel Kahneman also that also in the brain there's no free lunch. There's always a trade-off between the speed and the depth of decisions and solutions that our brains compute, which yeah gives rise probably to the two systems of Daniel Kahneman, thinking fast and slow. And with this, I thank you for your attention. Happy to take questions and then hand it over to Luca.
So yeah, my name is Luca, and I'm glad to do the second part of the session. So I will present the big topic of plasticity, and specifically how unregulated plasticity in Alzheimer's disease can be investigated with the virtual brain. So I started in this lab also as a master student. So this project slowly turned into a thesis and now is being distilled into a manuscript. So everything here is still unpublished. But nevertheless, I'm happy to discuss it with you. So let's start about defining what plasticity actually is. So the big question, how can the brain adapt itself in a dynamic environment? So how can from a finite set of structure rise these complex cognitive functions? How can we form memories? How can we learn new skills, new languages? And to all of these questions, plasticity gives the answer and the mechanism. And plasticity comes from plastic, so formable, and it's the ability of the brain to reorganize its connections in response to experience. And these plastic changes occur on many different scales, coming from the molecular side, where there are receptor dynamics, even genes regulating plasticity, up to the cellular level, which define how this synapses and the neuronal connections rewire and change their connections, up to the networks, forming distinct functional networks. And all of these plastic changes can then give rise to behavioral changes as well. So, for example, the recalling of a memory or learning of new skills, for example. So you might be familiar with the phrase neurons that fire together wire together, which came from Donald Hebb in the 1950s and was later coined by Löwel. So there's this whole dispute between some researchers who really came up with that catchy phrase. Yeah, so it describes the most fundamental form of plasticity pretty well. So if two neurons firing coincides, their connection strengthens. That's basically what it's saying. So two neurons being connected over synapse, and if the presynaptic neuron fires and the postsynaptic neuron fires too, there's some strengthening in connection of the neuron. So in future, the postsynaptic neuron becomes more responsive for future action potentials. So this is what we call Hebbian plasticity, coined after Donald Hebb. And you would have at least schematically similar networks in your brain for encoding the concepts of bananas and apples, for example, so you could distinguish between them. So there are some neurons encoding wait, let me activate the uh, laser pointer. Okay. So there are some neurons encoding, for example, the shape of the banana, the color, the smell. So if there is activation among any of these connections in the network, there would be activation of the downstream of the established connections. So if you would see a banana in the wild, you would be able to recognize it. Yeah, so this simple rule is the basis of networks being formed. And just to give you a short introduction in the molecular basis, so looking at the synaptical connections, what really happens on that scale. So Hebbian plasticity can induce long-term potentiation, so strengthening, and long-term depression, which is weakening of synaptical connections. So first of all, what happens when the connections strengthen? So these are again two neurons connected via synapse. So if the presynaptic neuron fires, there's some glutamate released from the vesicle into the synaptic cleft. And then if the postsynaptic neuron is depolarized, so there's coincidental firing, the NMDA receptors have a magnesium block that gets removed if both neurons fire together. And then calcium can influx into the cell. And if the calcium levels are sufficiently high, then AMPA receptors get inserted into the membrane. So if a future action potential arrives at the same connection, the postsynaptic neuron would become more responsive because there are more receptors in order to depolarize the postsynaptic neuron. And on the other hand, if there's no correlation in firing, so no firing together, their AMPA receptors get removed from the membrane because calcium levels are not sufficiently high. So the neurons become less responsive to their firing. So yeah, what I wanted to show you with this is that the NMDA receptors that are coincidence detectors basically are the molecular correlate of Hebbian plasticity. And this is what happens on the smallest scale, but we're looking into the network scale now. So I presented you what happens when neural activity facilitates changes in synaptic strength due to the mechanism of Hebbian plasticity. And these neural or the system can be influenced by external stimulation. And you can imagine that Hebbian plasticity is innately unstable. So, for example, if the stronger connections get higher neural activity as a response of increased synaptic strength, they would increase their connection strength further and further. So there would be like a positive feedback loop, which would cause overexcitation. So there needs to be something regulating this system, so to say. Um, and there comes in homeostatic plasticity, which is a regulatory form of plasticity. So depending on the activity of the system and the synaptic strength, it has different mechanisms to regulate the plasticity. So I really like this control theoretical view on plasticity here. So there are many forms of homeostatic plasticity, but just to give some examples, so synaptic scaling would be that there's some neural populations have a target firing rate, and the synaptic input is scaled towards the target firing rate. So if it's too high, then it's scaled down, or if it's too low, then it's scaled up. Um, yeah, metaplasticity, that's not you learning about plasticity, but it's a sort of a sliding window, so a varying threshold of plasticity. So when there were plastic changes in the past, the window would shift, so there couldn't be any more plastic changes, or not so easily. And then finally, inhibitory synaptic plasticity, which is responsible for balancing the excitation inhibition balance, for balancing the excitation inhibition in the network. And it's a similar mechanism of what Micha presented to you earlier. So if there's overexcitation, the inhibitory neurons get more active as a response of that. So they're linked together. So there's this seesaw dynamic between Hebbian plasticity and homeostatic plasticity. If there's enhanced homeostatic plasticity, the Hebbian plasticity would be altered, and vice versa. If the regulatory plasticity is altered, then this would lead to either increased or decreased Hebbian plasticity. And this is exactly what we're investigating now. So what happens when homeostatic mechanisms or neuromodulatory mechanisms are impaired, which causes unregulatory, or in our case, we term it excessive plasticity. Or in this sense, we mean excessive Hebbian plasticity, just to be sure with the terms. Yeah, so I explained you now a bit how plasticity can become unregulated, but now in the specific example of Alzheimer's, I will explain how this can also give rise to some cognitive deficits. So imagine you were watching a movie last night, so I don't know, maybe Inception, and there would be some memory traces, so some neurons encoding different parts of the movie, really schematically speaking, and you happen to be eating popcorn during that movie. So this morning you could recall most details of the movie successfully, but tonight you watch another movie, for example, um, I don't know, Spider-Man, and then again after Hebbian plasticity rules, there would be some strengthening of connections. And again, you ate popcorn today as well, so there would be some overlap between the memories. And this is really an important detail. And if plasticity now becomes unregulated, this leads to two distinct phenomena. So first of all, it leads to strengthening of the already established connections, because they were stronger and excited over the overlap of the network. because of the popcorn eating. And there would be some false connections between the two memories. So an excessive strengthening of the already established connections and false acausal, you can call them connections. So this is called a runaway synaptic modification and would lead to confusion between the two movies. And is thought to explain some cognitive deficits in Alzheimer's because patients are suffering from childhood memories being more present, like the already established connections being reinforced further, but new connections cannot be made properly. And Hasselmo came up with this nearly 30 years ago, and he did an experiment. So it was like a bit like a Hopfield network where they gave an input, and after Hebbian plasticity, there would be like an output of which connections are strengthened. And if there's interference during learning, it's exactly what I showed you here with the overlap of connection. So they interfere with each other. There's an excessive amount of connections being established, as you can see here in the dense clusters. And without this interference during learning, it is just as the input that was given to the network. And Hasselmo links it to neurodegeneration in Alzheimer's disease. So I will show you now. And in the healthy brain, there is cholinergic neuromodulation, regulating the synaptic transmission. So you could encode each memory separately without spreading between the networks. So usually you can recall both movies separately. Yeah, so what happens in Alzheimer's disease is that there's a loss of cholinergic neuromodulation, which we will focus on today. And this excessive plasticity can also be caused as a result of a lack of homeostatic plasticity mechanisms. So part of the same process, but this is the focus for now. And as you can imagine, this excessive amount of established connections lead to high metabolic stress because first of all, you need the metabolic resources in order to facilitate the strengthening of connections. And this causes a shift in the feedback and balancing system. So there's an overexcitation, even leading to excitotoxicity, like in this part, and also the metabolic stress due to this disrupted balance. And yeah, Hasselmo also links it to really pathology of Alzheimer's, so amyloid beta and tau, and that they appear as a result of this breakdown of the feedback mechanism. So tau is really involved in the transport mechanisms of the neurons, and they break down due to the stress and this tilted balance. And this then facilitates neurodegeneration and then further destabilizes the system. And this is what we investigate today. And one important network we will also look into is the default mode network. It consists of different regions, the medial prefrontal cortex, posterior cingulate, a region, inferior parietal lobe, and the hippocampus. And it was found in patients laying in the scanner address. So in the fMRI, they were told, so it's usually between experimental paradigms where they just fixate across. But as you might know yourself, it's really difficult to just not think about anything. So the people at rest, their function network, the default mode network, is involved in many different cognitive functions. First of all, self-referential judgment, episodic memory, so autobiographical memory too. So suddenly something, I don't know, from 10 years ago appears and I don't know, social cognition, thinking about other people. So it's called a theory of mind. and how they are related to use. So again, this self-referential judgment sort of, and language and semantic memory. So a lot of important cognitive functions. And notably in Alzheimer's disease, there's a decreased functional connectivity found in the default mode network. Also found in many studies, so also overall decreased functional connectivity. But in this study, they found specifically between the posterior cingulate and the hippocampus that there's a decrease in functional connectivity. So we are hypothesizing that it's somehow related to functional network changes, that plasticity is related to this functional network changes observed in Alzheimer's disease. I think by now you already are well taught about whole brain simulations and what ingredients are necessary in order to conduct such a simulation. But I will give you a short heads up. Yeah, Micha also already mentioned structural connectivity, and I stole his figure too. So we took DTI data from 1,000 subjects um, in order to construct the structure connectivity matrix. And from DTI, we take the amount of white matter fiber tracts. So and you apply a parcellation in order to use the fiber tracts as a metric of how strong the brain regions are connected to each other. So from that, we can create this structure network where the nodes are the brain regions and the edges denote the connection strength. And in this case, we apply the Desi-Kankiani parcellation, dividing the brain into 84 regions and normalize the weight from 0 to 1. And here you see the left hemisphere and the right hemisphere in the structure connectivity matrix. So for the brain dynamics, for simulating brain regional activity, we use the Janssen-Ritt model, and it is a neural mass model. So it's used for simulating cortical column activity. And the idea is in one cortical column, there's always interaction, or this is the most basic interaction in such a column. So there are two excitatory populations and one inhibitory interneuron population, and the excitatory pyramidal population have long-range connection to the other brain regions. So the brain regions can be coupled and can be stimulated. And on a bit more complex level of analysis, we don't have to go into detail here, but each of the each of the neuronal populations is described by two differential equations. And what we just need is that there's a coupling factor changing the input from each brain region. So how strong the whole brain dynamics, how strong the brain regions interact. And we will later stimulate and to see how the trajectory of plasticity changes and introduce neuromodulation by altering the global coupling. So this is all you need to remember from this slide. So from after having the brain regional dynamics and the structural connectivity that spans the structural network, the whole brain simulation can be conducted, yielding for each brain region simulated neural activity, which can then be transformed via Bolt monitor into blood oxygen level dependent activity. So it's a fMRI signal, if you want to put it into simple words. And from this Bolt activity, we can compute the functional connectivity matrix, whereas each entry of this connectivity matrix denotes how high the correlation between the other brain regions are. So for example, the the hippocampus with the prefrontal cortex in one entry. And so one would mean that we're completely synchronous and -1 anti-synchronous. So we have now all the ingredients to formulate a simple plasticity rule. So the correlation matrix or the functional connectivity matrix C and the structure connectivity matrix W. So after the Hebbian plasticity rule from the beginning, neurons that fire together wire together, we change the structural connectivity matrix by adding the functional connectivity matrix with the regulating learning rate. So this would mean if, for example, again coming back to this example of the hippocampus firing, having high correlation, having high synchrony with the prefrontal cortex, for example, in one column, then their connection would be strengthened as a result of that. So this is one plasticity iteration where the function connectivity is added on the structure connectivity. And yeah, we simulate for multiple of these iterations. So coming back to the idea, so I showed you how plasticity can become unregulated, specifically in Alzheimer's disease, how it can lead to different cognitive symptoms seen in Alzheimer's disease, and how we can integrate such a rule in the virtual brain, so in a whole brain simulation. And now we will look into the function network changes and try to relate them to the pathology seen in Alzheimer's disease. And then we want to stimulate the distinct regions to see if it alters the trajectory of plasticity and also apply neuromodulation. Okay, so again, this is this plasticity iteration loop. Here you will see the changes of the function connectivity matrix, the respective functional networks that were generated from this function connectivity matrix. Because it's similar to the structure network, you can also create function networks from the connectivity matrices. And here you see the mean of function connectivity over the different iterations. So it's a little bit of a spoiler, but I play it now. You see that there's an increase of function connectivity and then followed by a saturation. And looking at the function connectivity matrix, you see that there are some dense clusters occurring and then suddenly disappearing again. And among different modules, the connections are being created in the function network. So how can we investigate this? So again, the different snapshots before the plasticity iteration, there's a distinct division of the network into different modules here. And after simulation, you see that they are spread across the different modules. So the structure is sort of dissolved. So there's an excessive spread of connections. And so two things you can notice here is that, for example, looking at this node, you can see the node strength is by a higher color. So increased strength is shown by a brighter color here. So this node gets stronger and stronger over time. And interestingly, there are some regions that are disconnected after the simulation. And we will look into this later and why they are disconnected in the first place. So for that, we use a graph theory or network theory to understand what really occurs on that scale. So for that, three metrics are used. So global efficiency is a measure of how short the paths are within the network. So how fast can you travel, for example, from point A to point B? So like fast travel routes. Clustering coefficient is a measure of how divided the brain is into smaller groups, so to say. Like think of it as a social network. You're in the uh, dark red dot here, and you have a friend. So you're in a group. You have a friend, and this friend has another friend, and how likely the friend of a friend is to be your friend too. So this sort of division into smaller subgroups. And lastly, modularity is about it's called different communities within a network. So mostly they are functional units, or yeah, you can understand them as for example, in the city, how there are neighborhoods. So within a neighborhood, you would have smaller and stronger paths, and between the neighborhoods, there would be only sparse paths. So in order to create a function network, we need to threshold the graph, or threshold the functional connectivity matrix, because ranging from -1 to 1, there is like nearly an all-to-all connection. So to understand what happens, we need to cut out stronger, weaker connections. And for that, we apply a density threshold, whereas 15% density would mean the 15% strongest connection remain. And you can see if the networks become too dense, there's information about global efficiency and modularity altered. The clustering is not affected, but also networks being too dense generally loses information, and also too sparse. So then they might become disconnected and might not represent biological networks anymore. So we create a binarized graph from this. And after simulating plasticity, again, here over 300 iterations, there's at this peak of connectivity, there's also a peak of efficiency within the network, and then returning to baseline, an increase of clustering, um, which aligns with the smaller clusters that were like these sort of hypersynchronous clusters you were seeing in the functional connectivity matrix, and then this saturation phase, reducing the clustering, and the modularity is also reduced during this hyperconnectivity phase. Um, yeah, so how can we make sense of this? So actually the research about the function networks is really arguing a lot about how what really occurs in patients. So there are many different reported metrics, but I found the Breyer et al. Study to be fitting because so they investigated preclinical AD. So they determined from CSF, so the cerebral spinal fluid, if there are the markers. So I think the amyloid beta proteins are already present, but the patients did not exhibit cognitive dysfunctions yet. So they reported not affected efficiency, reduced clustering, and reduced modularity, whereas we only align with the reduced modularity. And even though it's a snapshot of the function networks of the preclinical AD patients, it could be linked to these plastic mechanisms, because we have this hyperconnectivity causing these increased clustering and also increased efficiency at a point. And this could be seen as a sort of a phase before that. So already causing the disruption in networks, and depending on which phase we look into, we find even overlap of the functional alterations they found. So it's really about the destabilization. So it would be interesting if even before that they find like this hyperconnectivity marked by by increased clustering, for example. Mm. Okay, so as I mentioned earlier, there are some nodes in the network being disconnected as a response of the thresholding. So here at iteration 70, plasticity iteration 70, and here at plasticity iteration 120, so at the peak of connectivity, of functional connectivity, there's some regions being disconnected. And then we ask, is there any spatial overlap with functional networks that were reported in Alzheimer's disease? And surprisingly, there is. So here are the most disconnected region. Disconnection rate is just a metric of how, if a node is disconnected, so it's binary. So if it's disconnected in an iteration, it's basically a count, and then so it 0.5 would be in half of the simulations, the region would be disconnected. And the most disconnected regions are the posterior cingulate cortical regions, followed by temporal regions such as transverse temporal, perihippocampus, and cingulate regions in general. And if compared to functional networks, they belong mostly to the default mode network, which is why I presented it earlier. So looking at only in 5% of the simulations, if the nodes that are disconnected belong mostly to the default mode network and some to the dorsal attention network too. But yeah, so at a higher threshold of disconnection rate, you see that they mostly spatially overlap with the default mode network. And then we can ask, okay, why does it why is it the default mode network? Does it have some intrinsic properties? And the first property that comes in mind is that the degree. So if you think that initially, so we look at the initial degree, so before plasticity was applied, if the degree is low enough, then it doesn't have so many connections to lose, so it gets more easily disconnected. And this is partially true. So there's a negative relationship between the mean disconnection rate and the initial degree. Right. And so the most disconnected regions, so the posterior cingulate regions, have a somewhat lower initial degree compared to other regions. But also this would mean that all other regions exhibiting a low degree would be disconnected at a similar level. So it can only explain partially that the default mode network is selectively vulnerable. And as you might know, the default mode network is also a hub in the communication of the brain. So there has to be something about the intrinsic properties or of the or intrinsic topology of the network of the default mode network. Okay, so now we're introducing cholinergic neuromodulation to understand if the trajectory of plasticity and of the functional reorganization changes. And for that, we introduce an a coupling, scaling factor for the global coupling, ranging from 0 to 1, whereas 1 would mean no change, and 0 would indicate a total disconnection of the brain regions, and introducing different levels. So the 1 again is the plasticity trajectory without modulation. So you see this increase in functional connectivity strength and then followed by the saturation. And because if we would have stopped after 300 iterations, we would see no changes in the other levels of modulation. So we have to extend the iterations, but also in order to account for a similar structural change, that is reached after 800 iterations usually. So what you see is that there's a shift in the peak of hyperconnectivity and among all of the levels. So you see the peak shifts, but it's also leading to some adverse effects in later stages due to the longer amount where plastic changes can accumulate. So partially we can say that cholinergic neuromodulation can lead to delayed effects of excessive plasticity and can also control the disconnection rate. So, or the disconnection rate and the disconnection, so the spatially selective disconnection. And here again, marked in brighter colors, you see the posterior cingulate regions, the hippocampal regions, and the disconnection reduces by increasing the cholinergic neuromodulation. Okay, so now you've seen cholinergic neuromodulation, so some alteration um, of the plasticity that could at least postpone and delay some effects, but not prevent them fully. And by introducing another level of perturbation, by introducing stimulation, so excitatory stimulation, we want to understand if it also alters the plasticity, the functional reorganizations. And there is a phonic stimulation applied in Alzheimer's disease, which is a deep brain stimulation, so an electrode that is inserted deeply into the brain of the patients, and electric pulses can be admitted to the region. So the fornix is connected to the hippocampus and involved in many functions of memory as well. So through phonics stimulations, they could alleviate some cognitive symptoms of the patients. And another study or another non-invasive stimulation that is not shown here, but we will look into is transcranial magnetic stimulation, so TMS. So superficial stimulation of the cortical surface by applying magnetic fields. And for that, so we stimulate two regions to simulate the somewhat therapeutic application. And for that, we pick the hippocampus, because in our postulation, the fornix is not spatially represented. So the closest would be the hippocampus. And for the TMS protocol, we pick the primary motor cortex because it's used in different stimulations, also combined with cognitive training that was reducing and alleviating cognitive symptoms or improving memory functions even. And what you see here is the disconnection uh, over the different function networks we were investigating earlier. And for the default mode network, here you see the control. The mean disconnection rate reduces by nearly 25% after stimulation. So this was also continuous stimulation. And in other networks, there were partially some increased disconnection as a result, and also some decreased. And we stimulated only the left hemisphere. And looking at the default mode network, the connections, the brain regions of the default mode network, you can see that mostly in the left regions, there was the biggest effect. So again, the disconnection rate in the cingulate regions here and in the parahippocampal regions was the greatest by applying stimulation. And plotted over the cortical surface, you see by a red color is a reduction in disconnection successfully in parts of the default mode network, and also some increased disconnection in the more occipital regions. So there are some adverse effects also occurring after the stimulation, as you already see in some of the networks. Okay, so I've shown you today is that the one of the fundamental mechanisms of plasticity can not be always or can become pathological when it's dysregulated. So the same mechanism that enables us all these important cognitive functions can become pathological and use the same mechanisms in order to cause different cognitive symptoms, especially as we've seen today in Alzheimer's disease. And we identified the default mode network as being selectively vulnerable to the plastic changes. So it might be linked to the disconnection also observed in the literature of Alzheimer's disease and the cognitive impairments related to the default mode network. And using TVB as a sandbox, we're introducing cholinergic neuromodulation and excitatory stimulation, we could reduce plasticity-driven disconnection. So we could postpone some of the plastic changes and even reduce some symptoms. And I hope I've shown you that just a really simple, a simplified mechanism can already enable us to generate a distinct hypothesis, and that we can explore within the framework of whole brain modeling. So it can be adapted in order to explore also the therapeutic avenue that are not as quite well explored already in Alzheimer's disease, for example. Okay, and with that, I want to thank you all, especially my shared first author on this project and supervisor, Leon Stefanowski, and other people that helped along the way and are also part of this project. And yeah, so thank you finally for listening, and I'm happy to take questions.